last time we were talking about the inner ear. Okay, and I'm going to show you guys another animation that's going to, I think, illustrate how hearing works. All right, so putting things in motion. And if you remember the working model of the ear that you guys looked at in the lab, so the little glass cylinder that you guys saw, that was supposed to represent the cochlea if you unfurled it or unrolled it. Okay, and so remember here, for hearing, we're looking at the cochlea. And inside the cochlea, we're going to have something called the organ of Corti. Okay? And so here is where we have hair cells that are attached to a nerve fiber that's going to send signals depending upon which part of the cochlea gets stimulated by a particular frequency. All right? Okay, so here, the animation I'm going to show you guys, or the video, you can find up on our Blackboard along with a bunch of other videos. Okay. It's this one here.
The sense of hearing is accomplished by a process known as auditory transduction. The ear converts sound waves in the air into electrical impulses, which can be interpreted by the brain. As sound enters the ear, it passes through the external auditory canal, where it meets the tympanic membrane. The tympanic membrane then vibrates in response to the sound. Sounds of a lower pitch or frequency produce a slower rate of vibration. And sounds of lower volume or amplitude produce a less dramatic vibration. Higher frequency sounds produce faster vibrations. The tympanic membrane is cone-shaped and articulates with a chain of 3 bones called the auditory ossicles. They consist of the malleus, the incus, and the stapes. The movements of the tympanic membrane vibrate the ossicles, passing on information, frequency, and amplitude. The 3 bones pivot together on an axis shown here in red. The pivotal axis is due to a series of ligaments which hold the bones in place within the middle ear cavity. The anterior palatal ligament and the posterior Equil ligament are of particular importance for the pivotal axis. 2 structures which normally obscure this view of the middle ear have been removed. They are the chordae tympani nerve and the tendon of the tensor tympani muscle. Through the ossicles, the vibrations of the tympanic membrane are transferred to the footplate of the stapes. The stapes moves with a piston-like action, which sends vibrations into a structure called the bony ladder. The ladder is filled with fluid called parallel. If it were a completely closed and inflexible system, the movement of the stapes would be unable to displace the parallel, and therefore unable to send vibrations into the bony structure. Due to the flexibility of the membrane called the round window, the stapes movement can displace the parallel, allowing vibrations to enter the ladder. The corridor leading to the round window is found within the spinal portion of the bony labyrinth, known as the cochlea. Vibrations produced by the stapes are drawn into the spinal system and return to meet the round window. The The portion of the spinal passage in which vibrations ascend to the apex of the cochlea is called the scale of vestibular. The descending portion of the passage is called the scale of tendon. A third structure, called the cochlear duct, is situated between the scale of vestibular and the scale of tendon. The cochlear duct is filled with a fluid called Endo, and when viewed in cross-section, the membranes separating through fluid-filled systems are visible. They are Reisner's membrane and the basal membrane. The membranes are flexible and move in response to the vibrations traveling up the scale of vestibular. The movements of the membranes then send vibrations back down to the scale of tendon. A specialized structure called the organ of Corti is situated on the basilar membrane. As the basilar membrane vibrates, the organ of Corti is stimulated, which sends nerve impulses to the brain via the cochlear nerve. The actual nerve impulses are generated by specialized cells within the organ of Corti called hair cells. The hair cells are closely covered by a structure called the tectorial membrane. As the basilar membrane vibrates, the tiny clusters of hairs are bent against the tectorial membrane, triggering the hair cells to fire. The entire basilar membrane does not vibrate simultaneously. Instead, specific areas along the basilar membrane move variably in response to different frequencies of sound. Lower frequencies vibrate the basilar membrane closer to the apex of the cochlea, whereas higher frequencies produce vibrations closer to the base. This arrangement is known as tonotopic organization. Together, this sequence of events is responsible for our acoustic perception of the world around us.
All right, dramatic ending. All right, so did that make sense to folks, what we just saw? So I'm going to talk a little bit about it, but it helps to see this in motion. All right, and I suggest you guys take a look at this. That link to this video as well as other videos are up on our Blackboard site. And so be sure to kind of put these things in motion. It really helps with understanding the physiology, how it functions. All right, so here with the looking at the hearing. So remember the way it was described is that we have a frequency which is going to be the distance between two peaks. So here higher frequency is going to be shorter distance between the peaks. Lower frequency is going to be increased distance between the peaks. So here higher frequencies, higher pitch, lower frequencies, lower pitch. Loudness is going to be the amplitude, how high these peaks are or how low the troughs are. Okay? So the height of these isn't telling you whether it's a low pitch or a high pitch, it's telling you how loud that particular pitch is. All right? And we measure that amplitude in decibels, loudness. Pitch is measured as frequency, which is in hertz. And so here, if we unfurl or uncoil the cochlea, right, and stretch it out, like the animation and the video was showing you, low frequency sounds are going to trigger here towards the apex of the cochlea. Higher frequencies are going to trigger here towards the, round, oval window, okay? And so one of the reasons for that is because of the basilar membrane here. The basilar membrane is kind of arranged somewhat like piano wires. So here, very short piano wires, or you can think about a harp. In order to make sure that you get deflection of that membrane, you need something that's high frequency. You need a little bit more force to do that. And so that's going to cause these hair cells to fire. Lower frequency, so if you have a kind of a longer string there, then you're going to get firing towards the apex of the cochlea. Now, each one of these hair cells that's in the cochlea, okay, is being mapped to a different part of the brain. So if these hair cells get triggered, then your brain knows that that's high frequency. These hair cells get triggered, that your brain knows that that's low frequency. And you put the combination together and you get hearing. Everybody following me on that? Okay. So the other thing is, how do we distinguish, say, sound versus, say, language, speech? Right? So that's something your brain has to discern. Right? Are there any questions on kind of how the cochlea works to map hearing of pitch and amplitude, so loudness? So, yeah, go ahead.
This is a little bit off topic.
Yeah.
I'm curious, a lot of these organs,
Mm-hmm.
are they regenerative in the sense that they break rock?
Some are better than others. Like we talked about with the olfactory neurons, you know, they do regenerate, but as we age, the regeneration ability decreases. So we don't replace as many of those dying, say, olfactory neurons, which is why our smell decreases as we age. You know, these are not as regenerative. And so these guys aren't nearly as regenerative. Gustatory receptor cells, you know, you can replace those. There's some stem cells to be able to regenerate those, you know, but these guys aren't very regenerative. Yeah.
So, in some cases,
Yes. Yeah, you're most likely going to be permanent. You know. All right, so the other thing I'm going to ask you guys about, all right, what types of receptors or what types of molecules or what was the mechanism by which, say, the olfactory neuron was triggered? It was chemicals, right? So we call those chemoreceptors, right? What about gustation? So think about the GPCRs that are involved. They were chemical chemoreceptors as well. So there's chemicals involved, right? What about the eye? Photoreceptors. Here, what do you think these types of receptors are? Mechanoreceptors, right? So we've talked about different special senses. Each one of those special senses has slightly different types of triggers for that receptor. So we got chemoreceptors, we have photoreceptors, and we have mechanoreceptors. All right, so just recapturing or repeating kind of what we talked about with looking at hearing. So hearing starts off with sound waves that are funneled in by the pinna or the auricle of the ear into the external ear canal, right, or acoustic meatus. They're going to cause vibrations of the tympanic membrane, which is going to transfer via the malleus incus and stapes those vibrations into the inner ear via the oval window, right? So like we saw in the animation, that piston pumping to move, transferring the waves of sound into waves of that fluid inside the inner ear. That fluid wave is going to be in the cochlea, and that's going to move particular hair cells depending upon the frequency of that wave. So higher frequencies are going to be closer to the oval window, lower frequencies are going to be further away. All right, so different pitches are detected in different areas of the cochlea because of this, right? So here, the loudness is going to be how many hair cells get activated by those mechanoreceptors. And then action potential is going down the cranial nerve that's involved with hearing and equilibrium. So this is one of the reasons it's called the vestibulocochlear, is because which branch of this is going to be involved with hearing? The cochlear branch, right? So does everybody follow the sequence of events here? Because you're going to want to make sure you know this for each one of the senses. Okay, so here's our vestibulocochlear nerve, cranial nerve number 8. Okay, and so that was the hearing, the cochlea. And where in our brain do we start having that processing that goes on? So here, the cochlear nerve is going to be passing information, eventually synapsing and passing information to a part of our brain in the temporal lobe. And if we're going to be identifying it, it's passed on as speech, right? So then we're going to be using Wernicke's area to actually interpret that speech language, right? So here, the processing that goes on, the primary auditory cortex, remember primary is always referring to that brain area that is actually receiving and processing the information. Association, right, is involved with what? What words do we usually use for defining what an association sensory area is? Interpret, identify, right? So here, that's what's happening here. Now, some parts are actually going to the midbrain for reflexes, right? So there's reflexes for in the midbrain. And if you have an emotional response, so music you like, music you don't like, what part of the brain's probably involved with that emotional response? What did we talk about for, say, olfaction or gustation? Limbic system. So anytime we have emotional memory and a response associated with it, part of this sensory pathway has got to be routed to the limbic system. You know, that's the system that's involved with emotional memory and so forth. Right? So remember that Wernicke's, it's really the association area for, say, speech. Right? So here we're talking about discerning what that speech is. Right? Interpreting, identifying. Is that clear to folks? Yeah. All right. So we're going to finish up with looking at equilibrium. All right, and so here we're going to leave the cochlea behind and focus on two other structures of the inner ear, vestibule and the semicircular canals. All right, so here, this is going to be mechanical senses that go on to maintain equilibrium, and there's really the two structures. The vestibules do static or linear acceleration, static equilibrium, and then semicircular canals are going to be involved with dynamic equilibrium or angular types of acceleration. Angular momentum there, okay? And so here are vestibules in this area, and then you can see our semicircular canals here. And at the base of each semicircular canal, there's a kind of a sensory structure. Now remember, the inner ear is filled with fluid. So here, vestibule and the semicircular canals are all filled with fluid. All right. So let's first talk about static equilibrium or linear. So I don't know why your book always puts these types of images with someone nodding their head, you know, because when you nod your head, you're actually using static and also angular, so semicircular canals are also being activated. So the better example of static equilibrium is if you're sitting or riding in a car and you accelerate or you stop or slow down, right? So your head's not moving, you're just moving in one plane, okay? In order to sense that, that's going to be static or linear, static equilibrium or linear acceleration, right? Okay? Also, if you go up and down an elevator, be able to detect that up and down motion without kind of turning your head, just keep your head straight, that's going to be static or linear acceleration. Is that making sense to folks? When you guys came into this room and you sat down in your chair, right? That was static or linear acceleration if you didn't move your head, right? Okay? When you get up from your chair, you're using static or linear accelerations being detected here in this area of the inner ear. Is that making sense to folks? Right? So here, how does this work? How does this system work? Well, there are, in the vestibules, there are structures that some sit vertical, like this, some sit horizontal. And there's cells who's got extensions. They extend into this fluid structure, and then on top of the fluid structure, there's something called otoliths that are crystals made up primarily of calcium carbonate. And so they're fairly heavy. So when you guys accelerate or decelerate when you're driving a car, right, what ends up happening is that this is going to move. The fluid kind of stays in a position, and it is going to, your body system is going to move in one direction, right? So it's kind of like if you had a bucket of water, and you're walking in a straight line, and if you suddenly stop, what happens to that bucket of water? Sloshes around, doesn't it, right? That's kind of what ends up happening. When you started with the walking of the bucket of water, if you walk really quickly, what happened to that water? It kind of moved towards you, right? So that's what's happening here with these otoliths and this otolithic membrane, is that the hair cells are coming up, and even though those hair cells, may be moving, the otolith and otolithic membrane don't move with it exactly. And so what ends up happening is that these cells have mechanoreceptors, just like what we saw with the hair cells of the cochlea. And so they send a signal that, oh, something moved, right, in this plane or this plane, right? So if these otolithic cells, right, are oriented instead of vertically like this, if they're horizontal, sticking straight up, and the otolithic membrane was here, that would detect movement in this plane. What about an elevator? That would be this example, right? Where you're going up or down an elevator, these otoliths and otolithic membranes aren't going to move exactly with the hair cells, right? They're going to lag behind a little bit. They're going to cause these hair cells to trigger an action potential that you detect as some sort of linear movement, right? Yeah, go ahead, Tyler. So if they only moved in one plane, right? So we're going to talk about the other equilibrium in just a second, but if their head never moved, if their whole body just kind of moved in one plane, forward, back, side to side, straight up, straight down, that's static or linear. Okay? Does that make sense? Okay? Which is why, when you nod your head, some part of your inner ear is moving up and down, but it's also angular too. So I don't like this example, too much. So you can see here that gravity moves the otoliths a little bit, but you're not just triggering, this type of sensory equilibrium. You're also triggering the angular part too, when you're nodding up and down and side to side, which I'm going to show you guys in just a second. But if this person was just kind of moving their head forward and back without any type of rotation, then we'd be triggering this type of equilibrium almost exclusively. Yeah. Yep. Yeah. Yeah, because they're responding to some sort of mechanical change. Yep. Right. Okay, so the other type of equilibrium is dynamic or angular. So I got this very sophisticated model to show you guys. You can tell I got the deluxe version because it has different colors, right? Okay, so the semicircular canals, what they are canals filled with fluid, and they're in each of 3 planes. And at the base of these, we have detectors, which is going to be this crista ampullaris with the cupula there. Okay? So what ends up happening is that you get, say, if you're spinning around or you're nodding your head, you know, side to side or up, down, that fluid, the fluid inside, all right, is going to not move as quickly as these receptors, which means that these hair cells on these receptors get activated. So it's the same thing with the bucket situation, but instead of moving in moving in a straight direction, what if you spun around with the bucket? Okay? Right? So when you started that spinning, the liquid in the bucket, right, didn't keep up initially. But what if you suddenly stop? What happens to the liquid in the bucket? Does it stop immediately? No, it still sloshes around, right? Okay? So let me show a video really quick of how this works, and might answer your question. If not, feel free to ask it after the video. So here we have semicircular canals. They're going to be able to detect angular movement, so up, down, and then side to side, right? And then around. Okay, so if you guys are out doing cartwheels versus backflips, right? So let me just put this, see if I can find it. There we go. So I think this illustrates what's going on. Oops. Let's forget. Okay, so question. Mm-hmm. 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 So what happens when you spin around and you suddenly stop? Does that endolymph suddenly stop? No, it doesn't. So here, what signal is being sent to your brain? Even though you stopped, it's saying that you're still moving, right? And so that's what's happening when you feel dizzy after you're suddenly stopping, is that liquid in there hasn't stopped and it's still signaling. Yeah, glad you asked that. Yeah. Go ahead, Tyler. Well, there's the cardiovascular, issue. Something cardiovascular is going on there as well. So you're losing blood pressure that's being detected, and your cardiovascular system has to catch up to reestablish blood pressure. So yeah, you're getting multiple inputs when you do that. Yeah. All right. Excellent questions, you guys. All right, why don't we take a break? All right, so I think we'll just tie up a few loose ends with looking at equilibrium here. So you're going to want to, just like with the rest of the senses, be able to go through the different steps that are involved with sending that information to the brain. So remember, head movement. So moving the actual fluid, the endolymph, which is going to trigger sensation from the hair cells. Vestibules involved with up and down, side to side, front and back. Okay, so acceleration, it's really in a single plane. The semicircular canals are involved with rotation, right? So if we have our semicircular canals and we rotate, that's going to be detected by one of the semicircular canals. If we tilt, right, that's going to be detected by another semicircular canal. And then if we rotate or tilt our head side to side, that's going to be yet detected by another semicircular canal, right? So any type of rotational movements being detected by semicircular canals. Is that making sense to folks? All right, so we kind of have two different equilibrium mechanisms or senses, ways to sense two different types of equilibrium. And remember, this is being routed through cranial nerve number 8. And that comes together with the cochlear part. So vestibular cochlear is referring to the equilibrium and also the hearing coming from cochlea. Now, where does this go to help with equilibrium? Well, there's a lot of other inputs that go in with maintaining equilibrium. So we're getting information from proprioceptors. So we're talking about Golgi tendon organs and muscle spindle organs, right? They're detecting stretch in tendons and muscles. Be able to maintain equilibrium. So when you guys walk up and down, for instance, stairs or walk along a stairway, right? We also have visual input coming in. And then we have the equilibrium input that we just talked about coming from the vestibule and also the semicircular canals. The vestibular cortex is found in the cerebral lobe, that's the insula, right? Cerebellum is also involved with integrating this information to maintain posture, reflexes, so things that you don't really think about for when you say are consciously standing on one leg or running and so forth, right? And so also there's going to be input from the motor cortex. So when you send out a plan to execute a motor movement, it's got to take into account all this equilibrium information coming from vestibules, semicircular canals, vision input, how much stretch is going on in your tendons and also muscles. So there's a lot of information that has to get integrated in order to maintain equilibrium. Does that make sense to folks? All right. Okay. All right, so motion sickness. What is it? So this is a common question that always comes up. And there's no exact answer. The best thought out there is a mismatch of sensory information. So if you're out on a boat in a cabin and the boat is pitching side to side, in front and back, and rolling, right? Your room, You don't have any windows. Your room, though, is not moving. So the visual input says that, hey, I'm not seeing this movement. So your information from your eyeballs is saying, I'm not seeing this movement. But you're getting movement from what area? What sensory area? From your ears, your inner ears, right? So those equilibrium areas, the semicircular canals, and also the vestibules, right? And so you get this mismatch of information. This is the best idea out there for what's causing motion sickness. So, yeah, go ahead. It's supposed to dull down, the brain. The ideas behind it is that it kind of puts the brakes on a little bit so that, you're not getting as much sensory information there. Yeah, it's kind of what it's supposed to, the idea out there for how it's supposed to work. Yeah. All right. Okay, so we're going to switch gears a little bit and get into the endocrine system next. So we're going to start off with just talking about a few terms. But it turns out that the endocrine system is, we've talked about some features of this already when we talked about the nervous system, when we talked about neurotransmission and specifically synaptic transmission. So what do we know already? Well, what are some glands that are involved with the endocrine system? Does anybody know any? Pituitary is a big one. A lot of times it's called the master gland, and we'll see why. Right? Does anybody know anything else about the endocrine system? The adrenal gland. Hypothalamus is intimately connected with the pituitary gland. We're going to talk about them a lot. The hypothalamus and the pituitary. Yeah. So, the adrenal axis, absolutely. Axes we'll talk about as systems. That's basically what an axis is. Absolutely. All right? And so you guys have some inkling of the endocrine system already, and we're just going to start off with talking about some of the other details here. Let's see. All right. So here, if we have our cell membrane, right? So remember with, say, neurotransmitters bound to a receptor that was on the cell membrane, right? Okay. So we had, say, neurotransmitter, and we had, say, a receptor for it, and it bound. Most neurotransmitters are water soluble. Does that mean that they can easily cross the cell membrane? They're water soluble? No, right? So remember that here, the cell membrane, most of that distance is made up of a hydrophobic portion of phospholipids, right? So what part of the phospholipid is hydrophobic? Anybody remember? Tails, right? The heads of phospholipids are hydrophilic, right? So here, most of this distance is hydrophobic, and most neurotransmitters are water soluble, so they couldn't easily cross the cell membrane, which means that in order for the information to get inside the cell from, say, a neurotransmitter, it had to bind to a receptor, which then caused a sequence of events inside the cell, turning genes on and off, turning proteins on and off, right? Okay. So it turns out that hormones, and we're going to define that in just a second, also fall into two classes. So unlike neurotransmitters, hormones fall into two classes. That's the way I should say it. All right? Some hormones are water soluble, meaning that the receptor for those water soluble hormones is going to be in the membrane, right? So here we can say water Water-soluble hormones, right, are going to have membrane-bound receptors. Everybody with me on this? Okay. Now, there's other hormones that are lipid. Lipid soluble. So if they're lipid soluble, can they relatively easily cross the cell membrane? Absolutely, right? So it turns out that where are the receptors located for these lipid soluble hormones? They don't have to be on the cell membrane. They could be inside the cell. So here we could have a receptor. Actually, let me draw a different shape for this guy. Right, inside the cell for lipid-soluble hormones, because the cell membrane is not much of a barrier to them compared to, say, water-soluble hormone. Everybody with me on this? Okay. So once again, once this hormone binds to this receptor, it can cause all sorts of effects in the cell. Right? Just like with the water-soluble hormones. Okay. Does that make sense? Difference between water and lipid-soluble hormones, and where are you going to find their receptors? Everybody with me on that? All right? Because we're going to move on a little bit and talk a little bit about some classes of chemical messengers. One's autocrine. Another one's paracrine. And the last one we'll talk about are going to be hormones here. All right. Okay. So auto, what does that refer to? So self, right. And that's exactly what these factors do. These chemical messengers. So we have a cell, and it is going to secrete a chemical messenger, and that chemical messenger is going to act on the cell that secreted it. So here, this cell should have what in order to respond to this messenger? Should have the receptor for it, right? So that's exactly what happens, right? So we got a receptor, and then the same chemical messenger that was secreted from the cell binds to the receptor on the same cell that secreted it, right? And then you get some effects. There's examples of this, like in the immune system. So, for instance, certain types of lymphocytes, when they encounter an antigen from a bacteria, let's say, they activate themselves by secreting a factor, and they have a receptor for that factor, so they get activated. Right? So there's examples of that in the immune system. So is everybody comfortable with what we talk about with this type of chemical messenger, this autocrine? All right? So basically, the cell that secreted it has a receptor for it, so it can respond to the same molecule that it secreted. All right? Let's talk about paracrine. All right, so paracrine refers to a cell secreting some sort of chemical messenger here, a factor, and cells nearby respond to it. if these cells nearby respond to it, what do they have to have? The receptor, absolutely. So the cells nearby, if they respond to it, have to have the receptor. Right? So here, what ends up happening is that these molecules get secreted from one cell, and then they're going to bind to the receptor. Give that one there, and then you're going to get effects in these cells that have the receptor. So intracellular effects within the cell. Right? So has everybody seen the difference between autocrine and paracrine? What does this paracrine kind of remind you of? Think about the nervous system. So one cell is releasing a chemical messenger, and another cell is responding to it because it has a receptor. So what would we call this kind of space here if we were looking? Synapse, exactly, right? So if you want to think about this, think about the neuromuscular junction, right, that we talked about. If this is a neuromuscular junction, what cell is this? It's releasing, or let's start off with what's this molecule representing? Acetylcholine. And what cell's releasing acetylcholine there? So it's a presynaptic cell. Specifically, we call that the motor neuron, right? The cell that's responding. What cell is that? It's got so what's the receptor here? If this is acetylcholine, this is the acetylcholine receptor. And what cells in the neuromuscular junction have the acetylcholine receptor? Skeletal muscle, right? So here we could say that neurotransmitters act in a paracrine mechanism, right? Because there's a cell that secretes that chemical messenger, and there's a cell nearby that responds to it. Does that make sense to folks? So neurotransmitters are an example of this paracrine mechanism. Right? You look a little puzzled. It's not a second messenger, chemical messenger that's coming out of here, right? So, yeah, so here, this is the first messenger. And then if we have some sort of pass off of information, then it's a second messenger. Okay? Yeah. That's paracrine. How is that different than autocrine? It binds, yeah, so it's stimulating itself. The same cell that secreted this is responding to it because it has a receptor. Yeah, go ahead, Tyler. Yeah, so the factor here, so we'll see it's not a hormone, but yeah. It binds to a receptor. Well, to activate itself, right? So here an example is, in the immune system. So T cells do that, where they are going to activate themselves. And there's also chemical messengers. They don't fit neatly into the categories we have for them. So some of them also act as autocrine and paracrine factors. So here we're just talking about different mechanisms. And so, here we generate categories and bins for biology, but sometimes biology doesn't neatly fit into those categories. So here we're just giving you some terms to be able to describe these. Yeah. So why it happens, a lot of times when you ask why, it's a little bit challenging. Scientists haven't gotten that far yet to figure out why it's beneficial to do it this way and not that way. So there's probably ongoing research to address that. All right, so last thing I wanted to talk about is hormones. How does this function? This is going to be really important for the endocrine system. So here with hormones, we have a cell that's going to be secreting our hormones. and it secretes them into the blood. So if this chemical messenger, this molecule, gets secreted in the blood, where does it go? Where does it go in the body? Everywhere, systemic. Yeah, it's going everywhere, right? Now, do all cells in our body respond to all chemical messengers, hormones? No. What do they have to have in order to respond? Receptor, absolutely. So here, even though this hormone's in the blood, some cells don't have the receptor for it. So are they going to respond? No. Some cells, I'm going to draw it down here, will have the receptor, meaning that they will respond. Right? So this is the definition of a hormone, is that here, these chemical messengers, these hormones, are released into the blood, and they are going to be systemically distributed, and only cells that have the receptor are going to respond. Everybody with me on that? Okay. This is different than neurotransmitters that we saw. Right? Neurotransmitters, did they ever get secreted into the blood? Right? So they don't get secreted in the blood, and they act in a very localized fashion, right? As where here, these hormones are going to go everywhere. All right? Is that making sense to folks? All right. So that's some of the basics for the endocrine to start our discussion. All right. So here we talked about what do we already know. We have water versus fat or lipid-soluble hormones. We have glands that are going to be releasing hormones, and then some hormones are going to have specific targets based upon which cells have that receptor for that hormone. What you guys are going to need to know are what hormones are released from which gland and what the targets and functions are for the hormone. And so lab this week is focused also on this. So lab time is also going to be used to help you with understanding what we're doing in lecture this week. All right? Okay. So here, structures of some glands and hormones. Where do we find them? Some hormonal structures. So we talked about the pituitary and the hypothalamus. Some other glands, as you'll see in lab, are the thyroid and parathyroid. And then we have also the adrenal gland, which is sitting on top of the kidney. And then we have the pancreas. So part of the pancreas has an endocrine function. And then we also have some gonadal hormones, the testes and the ovaries. And we'll talk more about those hormones when we get to reproductive biology in 233. All right. So we talked about autocrine and paracrine. Okay? We haven't kind of talked about endocrine versus exocrine. So we did talk about that with glands back in 231. Endocrine means that it's being secreted within the body, within the blood vessel system, right? Exocrine means that it's being secreted into a structure that opens up to the external environment. So what's an example of exocrine secretions? Sweat, absolutely. Now, don't forget that the GI tract also opens up to the external environment, right? So digestive enzymes that are secreted into the intestine, for instance, would be considered exocrine, right? So secretions that are kind of staying in our body and distributed systemically, those are going to be more part of the endocrine system, right? And so here, do all cells respond to all hormones? We've answered that question, right? They only respond if they have the receptor. We've talked about this also, water-soluble versus lipid-soluble, right? So which ones are going to have membrane-bound receptors? So I thought I heard it. Water-soluble, right? Which ones are going to have intracellular receptors, receptors within the cell? So lipid-soluble ones, right? Okay, fat-soluble. Okay, so here, water-soluble. A lot of these hormones that are water-soluble are peptides or amino acid based. And a lot of the hormone receptors, not surprisingly, are what type of receptor? Well, anytime I ask that, if you say GPCR, you're probably right? So they're G protein coupled receptors, which means now we get second messengers, right? So here, the first messenger was a hormone binding to the receptor, the hormone itself. Second messenger is going to be other molecules, chemicals that are made up in the cell from an enzyme. And a lot of times it amplifies the signal. Right? Now, what ends up happening is we can cause the second messengers, or the effect of the hormone binding to the receptor can cause channels to open and cause proteins to be turned on and off, and also can cause genes to be turned on and off inside the nucleus. Okay. So here's a way that we can exert control from outside a cell to inside of a cell, right? It's a way to communicate across the cell membrane. Okay. Right? Does that make sense to folks? So water soluble, right? They have a membrane-bound receptor, and many times it's activating a second messenger system that we've seen before. Cause genes to be turned on and off, and also proteins to be turned on and off. Any questions on the water-soluble proteins? Or water-soluble hormones, excuse me. All right, so this will be a last slide. Lipid-soluble ones, a lot of them are steroids, meaning that they are synthesized from cholesterol. So here's the structure of cholesterol. Am I going to ask you to draw this? No. Right? I'm just pointing out that here's the structure of cholesterol. Look how similar a lot of these steroid hormones are. They have very similar structure to that of cholesterol. Cholesterol, we know, is very lipid soluble. Not surprisingly, these steroid hormones are lipid soluble. So here, once again, if we have similar chemical structure, we're probably having similar chemical characteristics, right? Here, lipid soluble, therefore we have, or cholesterol is lipid soluble, so many of these hormones that look like cholesterol are also lipid soluble. Now we can kind of break these lipid hormones, these steroids, up into three broad categories. Some are called glucocorticoids, some are called mineralocorticoids, some are called gonado or corticoids or sex hormones. Where's the receptor located for these guys? They're lipid soluble. Inside the cell, absolutely, inside the cell. Okay? All right. So, like I said, that's the last slide. And we'll pick up this discussion on Thursday. So the extra credit activity I have you guys doing in lab is to help look at how these steroid or these hormones in general affect each other. All right? Okay. Have a great day and be safe out there.